Hello everybody, thank you for visiting my poster. My name is Sandra Gonzalez Sayer. I am PhD candidate from National University of Colombia from Biotechnology PhD program. In this opportunity, I will talk you about the significant size expansion phenomenon mediated by a specific transposable elements inside the Pseudocercospora genome. It is a work performed in cooperation with the Laboratory of Computational Evolutionary and Systems Biology from Sao Paulo University, and also in cooperation with the Laboratory of Evolutionary Genetics in the Neuchâtel University from Switzerland. As an introduction, it would be good to start telling you what natural rubber is and where does it come from. So natural rubber is a biopolymer that represents the raw material for the manufacture of more than 50,000 products belonging to the medical and automotive industries. So this polymer is naturally produced by Hevea brasiliensis tree. Unfortunately, and even though when the Amazon River basin is the diversity center of this species, Latin American countries are not competitive in the natural rubber global production. They just contribute with 2% of the rubber to the world. The reason of this low competitivity is a disease known as a American Leaf Blight, which is caused by the ascomycete fungus Pseudocercospora ulei. So this fungus is particularly poor study. There is not any molecular evidence about the biology and the pathogenicity cycle. Due to our main goal in this project is to know the molecular basis of the Pseudocercospora ulei life cycle and also define the molecular compound that govern its pathogenicity. To do that, we decide to assemble the Pseudocercospora ulei genome. So we use a hybrid sequencing strategy, and we generate then both short and long reads. So the Pseudocercospora ulei assembly were built only with Pacific Bioscience raw data. It was built with CANU software. Then we use Illumina data to perform the polishing, and finally we use the nanopore reads, the correct one, to perform the scaffolding of the genome assembly. So below this strategy, we generate a genome with 214 contigs with a M50 value of 2.8 megabases and with 97.5 of single copy BUSCOs as a completeness measurage. So it is very important to tell you in this point that Pseudocercospora ulei has the biggest genome so far reported to the Mycosphaeraceae family. Once we generate the Pseudocercospora ulei genome, we proceed to optimize the structural gene annotation, which were performed with Breaker2 software. We do that by adding evidence from different origins. So we use protein non-redundant database, RNA-seq data, and also orthologous genes coordinates from the Pseudocercospora ulei genome assembly. We use information from Pseudocercospora fijensis, which is the taxonomic closest species and constitute the black sigatoka disease in banana crops in company with Pseudocercospora musa and Pseudocercospora musa. The structural annotation result reveals that this genome harbored 906,045 genes with an average of 4 exons and 3 introns per gene. We also found a very low percentage of overlapping genes, They are usually related with natural antisense transcripts that used to be overrepresented in fungal genomes. This is maybe the most exciting results in my project. We deciphered that Pseudocercospora ulei genome has an exceptional transposable elements proportion, which is represented by 80% of the total size genome, and is occupied by LTR and unknown Ts mainly. So, on the other hand, comparing with the black Sigatoka species complex, we identify that this genome presents more diverse T families. Finally, we perform a more specific annotation, and we realize throughout it that this unknown content is overrepresented by non-autonomous class 2 transposable elements known as mites. I want to finish my presentation sharing with you the results of the Pseudocercospora ulei secretome annotation and effectors predictions, comparing with 3 hemibiotroph species. The results are giving to us hints that Pseudocercospora ulei could present biotrophic lifestyles. If you realize here in the carbohydrate active enzymes, it is almost the middle to those present in the genome of the other Pseudocercospora species.